It's my great pleasure to introduce now our speaker, last speaker of today's first session, Andrew Adey. He's from Portland, Oregon University, State University in the US. And for him, it's a very good morning, I would say, early on for you to give this presentation. For us, it's late afternoon. And we are eager to listen to you. Andrew is somebody who's already joined us physically in this meeting. And I think you enjoyed this as well as we enjoyed your presence and presentation and discussions in Saarland when you came over. So now we want to see how much progress you have made and what we can learn new from you. So please, the stage is yours.
Share. All right, how does that look?
Looks great. Everything's
Great.
fine.
All right. Well, first, thank you for the invitation again. I really did enjoy a couple of years ago when I was able to be there in person. I very much enjoyed just meeting everyone and spending time there. So it's unfortunate that it's virtual, but I'm really excited to be here again virtually and presenting. So today I'm going to be talking a lot about some of the technology components that we've really been pushing forward on in my lab. And what we've really been focusing on is kind of several areas, right? So in single cell omics, there's these different components that we've tried to move forward. One is scope, so what properties can we actually assay at the single cell level? Scale, trying to produce very large data sets to meet the needs of some of these large atlas type projects. Context, so actually capturing some amount of spatial information, and we have a way of doing this in a very simple, easy way. And then content, where we're trying to improve the amount of data and information content that we're actually obtaining per cell. So the method that we use for all of this is single cell combinatorial indexing, which at this point is much more common, and I think it's been around long enough that you're probably fairly familiar with it. So I'll go over it fairly briefly. But the way that this kind of platform works, and I'm describing the attack version, is we take cells or whatever tissue that we're working with, we isolate nuclei. We also have ways that we can isolate cells instead and permeabilize them. We distribute these to the wells of a 96 well plate typically, such that we have the same number of cells in each well, and it can also be split across different samples and different sets of wells. These nuclei are permeable such that we can actually perform tagmentation for attack purposes, where the transposase can enter into the nucleus and act on the native chromatin. So this is in situ tagmentation, which is now used for most of the platforms for single cell attack-seq, whether it's combinatorial indexing or not. So the 10x genomics platform, for instance, you're tagmenting intact nuclei just without index complexes, and then you load them on the instrument. In our case, we're actually using index tagmentation complexes. So there are adapters that have barcodes that correspond to each well of that 96 well plate or multiple 96 well plates. After tagmentation, these nuclei are still intact, and those library molecules are kind of glued in place by that TM5. We pool all of those nuclei together such that in that pool, if I had a 96 plex tagmentation, I can pull out a random nucleus and then have a random one of those 96 indexes. So if I were to pull out a second nucleus, there's only a 1 in 96 chance that it has the same index as the one that I just pulled out. So we can differentiate between the molecules of those two different cells based on the tagmentation barcode. So we leverage that probability and actually distribute these in a limited distribution to a new 96 well plate or multiple 96 well plates, and then perform PCR. So you can see the color coding combination that represents these specific indexes, either at the PCR stage or tagmentation stage, is unique for each one of these nuclei down here. So we can differentiate between the molecules from each one of those. And it's these two tiers of cell indexing that allow us to sequence thousands of single cell attack-seq profiles in a single experiment, and just using 96 well plate based methodologies. So initially, we really wanted to scale this up because typical experiments would, we could maybe get up to 10,000 or so cells reasonably. So we wanted to go a little larger, and with that, we started using automation. So we use a 8-tip variable dispense liquid handling robot to distribute nuclei to these source plates. So from source plate to a bunch of tagmentation plates, and it dilutes them to the appropriate number of nuclei per well, appropriate concentration. And then we use a 96-tip instrument to actually stamp tagmentation plates, the tagmentation indexing reagent into the wells of these plates. Doing this workflow, we can actually produce up to 100,000 single-cell ATAC-seq libraries in a single workflow. It's a little unwieldy, so typically, we're shooting more for 50,000, which is a decent number, I think, for a number of applications. And to demonstrate this, and actually the reason we really worked on the automation was for a project that we have that's starting to be wrapped up now. We're hoping to submit it, and we'll put it out on bioRxiv as soon as it's ready, where we're profiling the primary visual cortex and the hippocampus from 9 individuals. And we actually performed NUN negative enrichment. So our goal for this project was actually to focus on the non-neuronal cell populations, because so much is done to focus on the neuronal populations. We wanted to give glial cells some attention. In total, we produced almost 400,000 single-cell ATAC-seq libraries. As far as the quality in this assay, one of the things we've done a lot of is optimizing our assays to work really well on postmortem human brain tissue. We just really spent a lot of time focusing on that. And I think it's shown and proved to be successful because we've gotten pretty good TSS enrichment for all of these libraries, regardless of postmortem interval. So even individuals where the postmortem time is over a day, we're still getting a pretty similar TSS enrichment value, which is usually what tends to drop off. It's not the same when it comes to RNA. That tends to drop off with postmortem interval a lot quicker, but at least ATAC, we're able to actually preserve that using the methods that we have to preserve these tissues. As far as unique reads per cell, we got a decent amount between 25 and about 16,000 for the 2 different regions as the median or mean. Analyzing all this together, this is just a UMAP of all the cell types. This is with some of the methods that we were using weren't really working very well on this large data set. So this is just latent semantic indexing, but we're able to identify the major cell types, but really the focus has then been to identify the major cell types and then pull those off and analyze those individual cell types on their own. And I'm not going to get into detail on all of the results, but this is kind of a vignette for instance, of just what we found with astrocytes. There's a fair amount of inter-individual heterogeneity. So we did perform inter-individual batch effect correction. And we're able to identify kind of some major cell type groupings within astrocytes and identify major genes associated with those, some of which are known, some of which are I think some novel hits that we identified, which is pretty exciting. One of the more the vignette and the cell type that I'm actually the most excited about is one that's typically less interesting, which is OPCs, so oligodendrocyte precursor cells. These are typically very homogeneous, and in fact, we didn't observe really any inter-individual heterogeneity. All the individuals tended to cluster very nicely together. And then when we projected it into 2 dimensions with UMAP, which there's a lot of issues with projecting anything high dimensional in 2-dimensional space for visualization, but it just looked like a big blob, which is what you expect when there's high homogeneity between the cells. But we did identify 2 distinct clusters, actually, in the higher dimensional data, which was interesting. And there's clear markers associated with these, and we've actually identified a subpopulation of these OPCs that are actually starting to undergo differentiation, which to our knowledge in postmortem human brain tissue, this hasn't been identified yet, where there's actually a clear difference between these. Now, it doesn't look very clear in this 2-dimensional UMAP, but when actually visualizing it in 3 dimensions, it actually gave us enough separation to actually be able to see that this kind of tail here actually pulls out and gives us this nice grouping of cells here. We performed pseudotemporal ordering to actually get kind of the progression of that differentiation. So we're pretty excited about this, and also rendered it in Blender, of course, taking a page out of Cole Trapnell's book, if any of you have seen any of his talks, just to make it look fancy. But I think it's pretty cool actually being able to pull out this differentiating OPC population. So the next thing I want to talk about is it's published work, so I'm not going to go into too much detail on it, but thinking on the spatial component, we really wanted to find a way to actually capture some of that spatial information of complex tissues. And there are a lot of really cool ways to do that, where you're using, indexing oligos to spatially, capture that amount of information. And those methods are really cool, and I think that's where things are going to be moving. But we wanted something that was a little more simple and direct, and that could be done now without having to do much development. So more methodological kind of aspects. So we really actually, to develop this, we consulted a lot of pathologists to see kind of what they wanted and what made the most sense for moving some of this work forward. So with that, we developed the SkyMap attack protocol. And the way this works is pretty simple. We just take these cryosections, these sections, and then microbiopsy punch them. So we actually do 200 micron thick sections interspersed with thinner sections that we use for imaging. And then we use these microbiopsy punches that are 250 microns in diameter. So the total tissue voxel size is about 215 microns in diameter. And then we deposit each one of those into each well of a 96 well plate or multiple 96 well plates. And then we use the tagmentation index to actually spatially encode. So that allows us to give us this barcode. And we're actually getting single cells from each one of these microbiopsy punches. The big advantage, one, is that it is simple. You can actually do targeted region of interest selection, or just kind of stamp out a grid, which we're moving into robotics to actually do it with higher precision, higher resolution, and by developing that out. But that's kind of been on hold with the pandemic. But it's also very versatile. So we're able to use this for attack, or we're moving it into single cell DNA methylation as well, because it's really just this upfront tissue processing that gives us this spatial component, coupled with the high throughput sample indexing of combinatorial indexing. And just an example, we apply this to the human primary visual cortex. We're able to actually stamp and punch these trajectories through the cortex, through the lamination, going from the outer to inner cortex, as well as some kind of some white subcortical white matter punches. And then if we just look at the UMAP and color code the cells based on the category of the punch. So each one of these colors actually represents multiple spatial coordinates, but there's from the same category. You can see that we've got a nice mix here. These are oligodendrocytes, but in the glutamatergic neurons, you actually see some spatial gradient there based on that shading, which corresponds to outer to inner cortex. And we can actually look at cell type composition based on the ordering from positions 1 to 8. So this is actually looking from each one of these 215 micron voxels, what the cell type composition is moving through that. We can even take an individual one of these trajectories and analyze those cells individually. So this is just from 8 individual spatial punches, looking at the cells. We can do cell type identification. These are glutamatergic neurons here, and this kind of cluster. And we can actually then isolate those individual neurons. So just one cell type from one spatial trajectory, analyze it on its own. And you can still see that spatial kind of gradient, at least in UMAP space, actually persists from analyzing just this relatively small number of cells. And it actually matches up with some of the markers that we expect for having that outer to inner cortex kind of trajectory and those different factors that are associated with those different layers. We also apply this to a mouse model of cerebral ischemia. I'm not going to go into detail on this because it's published, but it was pretty cool. We were able to do this kind of plus pattern that kind of captured the gradient around this injury site, and we identified a variety of different factors. Most of them have already been known to be associated with stroke, but as well as some novel factors that are associated with this kind of the penumbra around the infarct zone. So the next major focus is, is really trying to get more information content per cell. So this is an assay that we've been working on and just recently published, and we're continuing to expand it into other properties. And it's based on, it's really only for tagmentation-based assays. And one of the things that has kind of bothered me so I worked on developing tagmentation just for original DNA-seq back as a graduate student. It's actually my rotation project in Jay Chendra's lab. And it always bothered me that you're tagmenting with random forward and reverse adapters. So half of your library molecules end up getting a forward and a forward adapter or a reverse and a reverse adapter. And those molecules are not viable for downstream processing. So you're losing half of your library. So your theoretical maximum is only 50% of the yield that you can obtain. And that's true for a TAG-seq or any standard tag-mentation assay. And we've tried various ways over the years to try and get around that, and it's worked with moderate success until recently when we worked on this new kind of approach we call symmetrical strand single cell combinatorial indexing, or just S3 to keep it brief, where we tagment with just a single adapter that has an index. So each index corresponds to each well of 96 well plates, that same combinatorial indexing concept. But it has this uracil present in the adapter right before that cell index, or that tagmentation index. So then after tagmentation, we pool, we sort them and distribute these nuclei into their respective PCR wells for the second tier of indexing. And we do this pre-processing step where we first extend the end out here into this mosaic end, but we're using a uracil intolerant polymerase. So it extends through the mosaic end, and this is kind of the universal tagmentation or TN5 recognition sequence. So it extends through that, but then it stops at the uracil. So it doesn't copy the cell barcode or the TN5 barcode or the forward sequencing adapter or indexing adapter. It just stops after this mosaic end. We then add in this other adapter that has LNA, so locked nucleic acid. This just has a higher affinity, a higher TM, and binds more tightly with that mosaic end sequence there. It's blocked, so it can't act as a primer, but it acts as a template for these library molecules to actually extend through and copy that reverse adapter. So now each one of these strands, the top and bottom strand, from each library molecule can actually extend out and be a viable sequencing library molecule. And we can actually do multiple rounds of that. So we do about 10 rounds of thermocycling to do that linear extension. So we get a lot of chances to copy that reverse adapter. And then we perform indexing PCR as usual. So this allows all of our library molecules to be viable as opposed to that 50% maximum. There's also some other benefits that actually convey increased PCR efficiency. The PCR is actually pretty inefficient for the first extension in regular tagmentation. So this kind of sets it up and actually gives us a substantial additional bonus to efficiency. So we did the kind of standard barnyard experiments, make sure we're getting true single cells. There's no cross-cell contamination. We like to do these experiments using primary tissue because it's just more close to reality than kind of cell line experiments where it's often, you often get good results compared to primary tissue. But this all looked good. I mean, this second one here is actually a control where if we see any single doublets or collisions, it actually indicates that there's barcode swapping during that linear amplification step. So it was a good control to make sure that that's not messing up with any of the index swapping or things like that. So this all looked good. And what was really exciting is that we got a very substantial boost in the log 10 unique reads per cell compared to other platforms. It's about a 10-fold improvement over our regular SCEattack assay. And then compared to droplet single cell TAG-seq or 10x single cell TAG-seq, we're over a 6-fold increase in the number of unique passing reads per cell at a comparable level of sequencing saturation. And this doesn't sacrifice TSS enrichment or any of the attack signal. I will say we highly optimize this for brain tissue. We recently did a prep on PBMCs, and the TSS enrichment was a lot worse. So, but I think that's primarily in the sample prep side. The way we do our nuclei isolation and all of that, we've just highly optimized for brain, and we kind of use the same workflow for frozen PBMCs. So that's something that we need to work on. But at least in brain, we get really high read counts per cell with a high fraction of reads and peaks. We didn't produce a whole ton of cells because just some aspects of the workflow, having a whole set of 96 indexes versus some advantages we can have with regular combinatorial indexing. But we did get very clean cell type separation, largely due to the high coverage we're obtaining per cell. And we're able to even do subclustering and break the interneurons into 4 different subclusters that match all the canonical markers of these CG and MGE interneuronal subtypes, including separation between those with like SST neurons and p-valvular neurons, or interneurons. So we were able to really identify the expected major cell types with high quality. We then extended this to perform whole genome sequencing, and this was using our workflow to do nucleosome disruption that we had previously published, and we're able to do basically what we do is open up the entire genome for tagmentation, and then it allows us to get whole genome sequencing. We also had some improvements to the nucleosome disruption, and this ended up giving us over 100-fold boost in our unique passing reads per cell, up to over 15 million unique passing reads per cell for single cell whole genome. So it's finally getting to the range where we can do real meaningful copy number calling. But we also performed one where we did this restriction digestion and then religation. So some portion of our reads will actually be chimeric reads from long-range interactions, much like Hi-C. And we were able to get a substantial boost over the prior Hi-C assay, about a 17-fold improvement. And the log 10 contact, or the total number of long-range contacts per cell that are defining those as over 50,000 base pairs range. And we're able to get nice compartment calling across these. This is a pancreatic cancer, a patient-derived cell line with very low passaging to enrich for the epithelial population. And we're able to identify a subclone specific translocation using this long-range interaction data. The last thing I wanted to touch on is improving throughput. So this is one that we've had in the works for a while, where we've started shifting from using 96-well plates for our indexing PCR into using this 5184 nanowell chip. So these are 300 nanoliter wells using the Takara isolate instrument. So for this experiment, we did 9 tagmentation plates, and then so 864 indexes, distributed them to this indexing chip, and we're able to produce over 460,000 single cell ataxia profiles from a single experiment, where the total cost in reagents to produce this library was around $2,000 US. So extremely cheap. The bulk of that cost is actually the isolate chip, or the most expensive component anyway, which we can actually reuse. We reuse these chips about 5 times before we discard them. So that actually can be reduced based on the cost. Though sequencing is still expensive at the time, because the NovaSeq S4 is around 16,000. So the sequencing is by far the most expensive component. But we were able to identify all the major cell types. One of the marks, I think, of a good prep for brain attack data is being able to identify clean endothelial cell populations, and we got that using FLT1. There's a nice clean marker gene for that, so we were excited to get a very clear, definitive population of these endothelial cells. So that was nice. Unfortunately, with this prep, we had relatively high ambient chromatin. So this is why I recommend to everyone always spike in a cell line or a small portion of cells from a different species, because you can actually start to assess whether you have cross-cell contamination or what your potential doublet rate is. And this was relatively high. I mean, the standard is you want to be below 5%. That's kind of what's typical for like 10x and other assays, but we were a little higher. And that's because with our regular SCEattack, we actually go through this sorting step, and we weren't here. So we did extensive optimization over about a year, and we're able to get that down below the 5% level. With the extra washing, we lost some cells, but we were still around 225,000 single cell ataxic profiles. And then we've actually also extended this to a design that's similar to S3, and it's even cleaner, though the throughput is a little lower, though I think we can improve that pretty readily. So just comparing the throughput, regular SCEattack was, around here. With these nanowell assays, and this is log 10 cells per experiment, we're getting really high. And actually this one here, this latest one, the version 3, I think if we use 2 tagmentation plates and optimize some of the loading, we can get up to 100,000 cells. But what was really exciting is with all of this improved washing, the attack signal is much, much cleaner. So the mean fraction of reads and peaks is over 80%, approaching 90% in one of these. So this is just extremely high for ataxic data, meaning it's extremely clean, high quality data. But what was also exciting is the mean unique passing reads per cell just was even better than our published S3 attack data, where we're over 400,000 unique passing reads per cell. And the vast majority of those reads, almost 90%, are actually in peaks. So we were very excited about this new data set. And then also, just with even the 20,000 cells, we were able to do real clear clustering and even subclustering of these interneurons here to get a very large number of clusters. So we're really excited about moving that forward, and hopefully that'll be a preprint fairly soon as well. So with that, I want to acknowledge those involved, and I'll take any questions.
Thank you very much, Andrew, for this mind-blowing talk, telling us, well, your super duper technical advances over the last 2 years, I would say. Amazing stuff. And yeah, I had a question.
Hi, Andrew.
Hi.
Hey, great talk. I have two questions. One is, how applicable is the your new technique to bulk ATAC-seq? And if it is, I think you said that it was, and how it compares in simplicity to the actual cell processing protocol of, ATAC-seq as published by Jason and Howard Chang. Because I know your lab is a methods lab and you're extremely good. But in terms of usability, I was just wondering.
Yeah, so that's a couple components. So like with the S3 attack version, for instance, like one of the issues with scEpigenomics is that it required custom sequencing primers, custom sequencing chemistry, all of that. And that would, makes it very hard for other people to implement. With the S3 version, it actually uses all standard sequencing primers. Like you just run the assay and it's ready to go on a regular sequencer. And there's potential ways for it to be accessible more broadly that I can't get into details, but so I'm excited about that. That's one of the things we've tried to focus on more lately is making things more usable. Like we're often publishing these proof of principle technologies that we're excited about, but they're not really readily implemented by a user base yet. So we're starting to try to move, and I think a couple of our next things that we have coming out are actually focused specifically towards that. One of the major ones being DNA methylation, because our SCEmet assay for single cell DNA methylation is just not easy, and this new version we have actually is. So we're excited about that. As far as bulk attack, one thing that we found is if you actually treat bulk ATAC-seq like single cell, and the key component is actually if you just do identify a nuclei suspension, do tagmentation, and then sort those nuclei, get rid of all the debris, and then do the PCR on that, it actually produces a much cleaner bulk ATAC-seq prep. The main reason is that you're getting rid of that debris that would normally be in the PCR mix. So sorting just nuclei post-tagmentation just with DAPI, and then sort, maybe 30,000 post-tagmentation into a well, and then take that, clean it up, go directly to PCR. It actually produces much cleaner data. So that's a pretty easy thing to implement that I think has been valuable for us.
And can I ask another question? Do you have additional PCR steps compared to the normal protocol that may bias the reads or affect the reads in any way?
So every single PCR reaction done in my lab is done on a real-time thermocycler, where we actually monitor the amplification. And as soon as it starts entering exponential amplification, we stop the reaction. So we do the minimal number of PCR cycles for every single prep that we do, just to try and limit the number of duplicate reads, and that you can get a bit of a reduction in complexity if you go too high. And we found as soon as you're, maybe 2 cycles into that start of the exponential amplification based on Cyber Green, you have enough library to quantify and sequence, but you don't over amplify.
Cool.
So
thank you so much. Thanks.
Okay, thanks for the question. Gonzalo has a question as well.
Yeah, Andrew, I was curious about these 2 different OPC populations that you are finding. I was wondering if, because you are collecting the non-neuronal fraction, right? So I was wondering if one of the populations could be more like neurostem cell-like, and not, do they express like SOX10, PDGFR alpha, like the canonical OPC markers, or could this be like more a neurostem cell population, or even like Arnold Kriegstein published recently a paper on and where he defines pre-OPCs during the the second trimester of human development. So I'm wondering if you compare the profile and if they are similar or not.
Yeah, no, that's a really good suggestion comparing to those. We don't see the markers of early neuronal differentiation in those. I'd have to look and see. I don't have the list of like the top hits that we have handy, but it's markers of like let's see. I think one of the key ones we found was SP1R5 and MAG. So yeah, MAG. So beginning myelin production, starting that. So it's definitely in that OPC population going towards the oligodendrocyte route, right? Is that what you're?
Yeah. So no. Okay, but so then
other populations.
Yeah, because I mean, we at least in mouse, we have defined that there is like the committed OPCs, which are like OPCs that have already committed to differentiate and that start expressing like some myelin genes, like you mentioned MAG. So is that? But from your data, it seemed that you had like that was the major population, and then you had like a subpopulation that seemed to be a little bit more immature. Did I misunderstand that?
The larger population was immature.
It was immature.
So those ones are the more progenitor like, and then the smaller population are the ones that are exiting that into more differentiation. And all of the all these samples, the age ranges from like late 40s to early 60s, I believe, is the age range of these individuals.
Okay. Thanks.
There's a question by Anna.
This was great, Andrew, really fantastic. I was curious of a very technical question about in your most recent technologies. You said that most reads are now falling in peaks, and I was curious in terms of numbers of peaks. So are you saturating, increasing, decreasing, and also are the peaks the same between the different technologies, or are there some interesting biology actually in underlying the different things you capture?
Yeah, the large majority of them do overlap. I think in the data set there was only 20,000 cells, but we sequenced it very deeply. It was close to 1 million peaks that we identified. And that's actually, so in the data set that was like the 400 something thousand cells, we identified over 1 million, but some of those peaks seem like they're spurious, and we're doing some filtering on that, I think due to the cross-cell contamination. But the really clean data set, yeah, it was close to 1 million. Most of those overlap with annotated known peaks, either from like the ENCODE project. So we've cross-referenced with that, as well as just other data sets that we have or publicly available data sets. So they're largely matching up. They all look to be very high quality. But there are a lot of them that are new, and that's something that we do want to explore further, and just do the due diligence and make sure that they're true peaks and they're not any type of artifact. But we do filter out, blacklist regions and things like that. So it's not those. But yeah, we're excited to see what we can get with just this higher coverage, and really looking at those peaks really at the single cell level and getting a cell type specificity that we can do.